Hi, everyone, and welcome to today. Welcome to today's Seed World Innovation webinar. My name is Mark Zinkowitz, and I am an editor with Seed World, and I am happy to be your host. Today's topic is brought to you by Computomics, and we will be discussing harnessing the power of machine learning for a breeding program. Now, during today's webinar, you'll likely have some questions for our speakers. Please type these into the chat box at any time during the webinar, and we'll address them during the Q&A session that we will hold at the end of the presentation. Also, today's webinar is being recorded and will be made available at seedworld.com following the event. Today, we will hear from two speakers. Ruth Mays is Director of Global Business Strategy for Computomics based in Wales. Ruth has a scientific background with a broad knowledge of applied research in genomics and the life sciences industry. And she also has experience in organizational leadership, building and managing dynamic teams across multiple sectors. Dr. Keith Ruffiner is corn breeder at Beck's Hybrids based in Indiana. His experience in plant genetics and corn breeding, together with his passion and curiosity, significantly advanced Beck's corn breeding program. Keith develops commercial hybrids, always having the success of the farmer as his primary goal. Welcome, Ruth and Keith.
Thank you, Mark, for the great introduction. As Mark mentioned, I'm the Director of Global Business Strategy at Computomics. Today, I have the pleasure to talk to Dr. Keith Ruffiner from Beck's Hybrids. Keith is going to explain how he implemented our machine learning technology, Exceed Score, into his hybrid corn breeding program. Before that, I'd like to provide a brief introduction of the company. Computomics was founded out of the Max Planck Institute of Developmental Biology in 2012. Our aim is to bring new and innovative bioinformatic and machine learning technologies to industry. We are the only company using and applying machine learning for predictive breeding, and we've been successfully doing this for many years.
Keith, how are you today?
I'm good, thank you. I'm pleased to be here and looking forward to a great webinar here.
Okay, so
excellent, and welcome, Ruth. Before we get so I was just going to give a quick introduction and then I'll turn the floor over to you guys. I was just going to mention to our audience that our agenda today is split into 6 parts. So we're going to start with an introduction to Computomics and its machine learning technology. We're going to discuss the importance and utilization of a robust data set. We're going to talk about using the data to build population structures. We're going to then discuss simulation of all possible crosses using this technology. Then we'll talk a little bit about applying machine learning to a breeding program. And finally, the predictive power of machine learning technology. So we'll start with our introduction to Computomics and its machine learning technology. I'll turn the floor over to you now, Ruth. The floor is yours.
Thank you, Mark. Thanks for the great introduction. And as Mark mentioned, I'm the Director of Global Business Strategy at Computomics. So today I have the pleasure to talk with Dr. Keith Ruffiner from Beck's Hybrids. Keith is going to explain how he implemented our machine learning technology Exceed Score into his hybrid corn breeding program. But before that, I'd like to just provide a brief introduction to our company. Computomics was founded out of the Max Planck Institute of Developmental Biology in 2012. Our aim is to bring new and innovative bioinformatic and machine learning technologies to industry. We are the only company using and applying machine learning for predictive breeding, and we have been doing this successfully for many years now. So let me show you how the Exceed Score technology works. So we developed a technology that can considerably improve the prediction accuracy for complex oleogenic traits, such as yield. It differs from statistical methods, which generally use a kinship matrix approach. Statistical models, such as BLUPs, presume that every genotypic variation is equal and additive. Machine learning technology evaluates the influence of each individual marker and of combinations of markers that affect the phenotype. Exceed Score recognizes differences in marker effects at the genetic level. It also weighs and balances its predictions to take advantage of these effects, which include non-additives like recessiveness, dominance, overdominance, and of course epistatic interactions. These cannot be modelled using BLUPs or genomic selection methods. We understand that the environment plays a major role in influencing the phenotype. Our machine learning technology incorporates very heterogeneous data sets such as precipitation, wind speed, daylight hours, and many other environmental observations. We also optimize for multiple traits simultaneously, and we are the only technology that can really make sense of complex, multiple biological data sets. So it all starts with robust data. Building robust data sets allows breeders to understand their germplasm and to use the genetic information to identify a target and breed towards that target. Machine learning requires both genotype and correlating phenotype data, which includes a subset of both high and low performers, and should include samples from the complete performance spectrum, as the model needs to learn to differentiate between all of these. The initial data set needs to fit under an umbrella of genetics, which takes into account the marker parameters. The initial model should consider identity by descent versus identity in state. Machine learning can incorporate data sets of environmental factors, and ideally measurements should include multiple locations and multiple years to be able to predict for specific climates. I'll now hand over to the expert, Keith. Keith, can you please explain how you structured your data sets to optimize your testers to build robust populations?
Thank you, Ruth. My introduction to Computomics was a breeder looking for new tools. I'm a commercial breeder. I'm the guy that has to go and apply all these new technologies and breeding approaches and find the ones that can help me do my job better. And so when we were approached and had the initial discussions with Computomics, we decided that machine learning was a technology or an approach that had great promise and great fanfare coming into our world. And we decided that we wanted to explore this as a potential tool for breeding, commercial breeding. And so we decided when we discussed this that we would start simple. And simply, we said, okay, we want to predict hybrid performance. Now, a breeder does this in every time he crosses two inbreds together and goes to test them. That's what we do because our product is a hybrid. And here was a methodology of where we could predict performance of hybrids before going to the field. So we sat down and we said, okay, we're going to start very simple. Two parent populations. In our case, one was a stiff stock population with two parents and the other was a non-stiff stock, because those were the two pools that we could cross together to make commercial type hybrids. We optimized our measures because really we wanted to try and see what does this technology provide? So we wanted to provide a data set that was both balanced and exposed the genetics fully. So we optimized the way we measure our traits. We defined our traits. And then we wanted to sample the population uniformly. Think of two bell curves on both sides of the hybrid combination. We wanted to sample those so that we did not introduce any bias into these predictions or the model building. And then we did break a little bit from traditional breeding in that we tested this material in its entirety in multiple years in multiple locations, meaning that generally speaking, we test 1 year, we advance only the best, and they only go to the field to be tested. We tested everything to generate a nice data set. The two goals we had going into this were we wanted to generate a phenotypic database that exposed the true genetic and expansion of the populations towards the identification of commercial type hybrid combinations. Also, though, we wanted to evaluate and begin to learn more about machine learning and how to apply it. Our vision of machine learning at this point in time was not clear. Once we had the phenotypic data and the genotypic data, we provided that information to Computomics, and we began our discussion with our Computomics statistician, telling him what our goals were and what we wanted, what traits we were looking at. And this turns out that this was a very important step in getting towards the success that we eventually saw. One of the things that struck me, and as now the first question I ask when I submit a project and it's ready to come back to me, is they gave me a complete overview of both the phenotypic data and its quality, and then also the genotypic data and its quality. We have a terrific marker lab here that do a terrific job, but it's really nice to hear that feedback going into it, which basically said to me they were aware of what they were going to do and what they were working with. It makes a very big difference as you move down. The results came back in the form of a list of hybrid predictions. And what we were easily able to do is go and sort this. We asked the questions, what are these values? What do they mean? And essentially, they were breeding values of performance predictions of the hybrid combinations, all possible combinations in our study. We are applied people. We took those hybrids into crossing blocks straight into testing in the next year. But because we wanted to learn more about machine learning and such, we started to break the data down. And we realized very quickly that we could identify the elite progeny lines in both pools. And when we discussed this with our statistician, he said, I can do that for you and make that a part of the return. And that's what today is now. But what we were able to do is identify those progeny that had the highest potential as individuals in their pools. And that opened the door for us to do a lot of neat things to pursue this. So the first thing we did then was we made two new groups of hybrids. The first group is a point A to B commercial type, the highest ones already done. But what we wanted to do then, and this turned out to be a very important thing, is we wanted to validate the model. So we made a set of hybrids that was strictly to validate, take to the field and validate the model and make sure that the accuracy was there and what we expected. It was somewhat painful for a commercial breeder, which has a limited resource, in the fact that we included not only some from the very high ones, but a few in the middle and even a few on the lower tail. Now, as a commercial breeder, to use my resources to put out hybrids that are predicted to fail was a bit of a pill to swallow, but it's a very important thing that we learned at the end of the day. We also then made a second group that was really about trying to understand what machine learning found in the potential of these lines. And that is to say, they have high combining ability. Now let's find out whether it's specific or general and how they could be used. And what we did was we selected lines that had a relatedness to our study, but were not in it. So we had a higher coefficient of covariance. And then we also selected lines that combine well with the certain types of genetics. This is what breeders do intuitively. We call them swags, but in this case, they were very much focused and directed. What we found, though, was that instead of looking for results of things in the field, we found ourselves that we are now facing forward, putting things together with the anticipation and the predictions And so this was really a change in our way we approached the breeding. What we learned from this initial study was that good sound phenotypic base is critical. Take your time, do it well. Model accuracy is everything as you go into the future. We found that machine learning was much more powerful, and it's not what we thought it was, but whoa. In our case, we were limited, I think, in this initial study by the genetic potential. And I think we were being limited by the simplicity of the questions we were asking, because I think that's the key of machine learning. It can do amazing things if you ask the right questions. The other thing is that know your direction, because immediately we were presented with opportunities to breed and go move this forward. So knowing where you wanted to go in the future with this material was critical to responding appropriately and quickly. And then again, it's an obvious one, but it really needs to be mentioned, is working together as a team, whether it be breeders within our group or and especially with the Computomics statisticians. They can help us ask better questions because they're the guys that program the machine learning to do this. And then also we found that we need to eliminate intuitive thinking. Breeders are a close group. We have jargon, we have acronyms that mean nothing to a statistician. And by conveying our questions that we want to ask in a form that they can understand how to integrate them is key to success in the future.
Thanks, Keith, for the overview. I just want to sort of backtrack a little bit and sort of look at the simulation process. So parental candidates, as you mentioned, are based on previous year's performance or the breeder's intuition. And a low number of possible hybrids can be explored. With machine learning, you can explore every conceivable cross by simulating all crosses. So this is sort of demonstrated by the graph on the left, where you have a sort of kinship matrix statistical model, where you're identifying your top performers from a subset of your data. But as you can see by the graph on the right, you are also identifying your top performers or your general combining ability. But with the machine learning, you're also picking up specific combining ability, which is quite unique. So Keith, can you explain a little bit more about the simulation process and how this impacts your program? And of course, the selection process for the next cycle.
Sure. And this is going to be kind of a walk through the history of our projects here, but it's also an indication of the learning curve that we started on and where we got to. So our starting project with Computomics, there were actually two that we submitted in this. And the idea was to begin to understand machine learning. The first one generated 30,000 virtual hybrid predictions. And the second one, 50,000. We thought this was incredible. But when we went and we validated and we took the material in a commercial application to the field, we were astounded. The accuracy of the model and the ability to put those hybrids in the field that actually would advance. We did a similar side project where we did a more traditional prediction of general combining ability type of thing and made a set of hybrids just to compare what machine learning versus that could do. Machine learning's in a different league. Our advancement rates under commercial type conditions and selections was astounding, like nothing we'd ever seen before. What we were left with after this initial study, or these the two projects, was we knew we'd only just seen the tip of the iceberg of the power of what machine learning was and could do in a breeding sense as a tool. We started looking for ways to unleash more power. So simple things like we could increase the number of progeny in each population to give us more combinations. We also started to say, well, could we integrate genotypes that are under a genetic umbrella? That is to say, they have a high coefficient of covariance. They're related, identity by descent, not by in-state type of things. And This would give us a better or broader genetic diversity, greater genetic depth in our population structure, thus sort of giving the machine learning more to work with. And that's what we did in our second project with you. The next project generated 1.3 million hybrids. That sounds really great. And anytime you get over a million, you're really excited until you realize there are new challenges that you have. A list of 1.3 million hybrids doesn't fit in an Excel sheet. So you have to start to understand what you're going to receive back and how you can handle it. We took some measures in this one. We looked off what we learned off the first one. We increased the progeny in the pools. And in one case, we basically tripled the number. We also upped our chip number. We tripled the number of markers, giving the machine learning more information to work with. And then we basically selected populations, especially on our stiff stock side, that we seem to think had much greater genetic potential. That is to say, let the machine learning reach higher towards commercial potential with it. And then we did structure in genotypes. What was interesting is that the amount of testing to set up this project was equal to, or even maybe slightly less than our original, but the number of hybrid predictions, like I said, was 1.3 million. And so the field resource was reduced, but the information and the genetic gain was increased by managing these aspects to do nothing more than allow machine learning to go in and do what it needs to do. Besides the new challenges, the results of this, when we took this to the field, were just astounding. And what we found when we started to break this data down was that we could start with these numbers and a number of combinations. Machine learning gave us some indications of new and higher level interactions that opened up for the second set of questions we could never ask before. When you're working in populations of 100 or 200, you can't ask questions of higher level interactions that you can when your virtual population is in the millions. And this set of questions that we now started to digest and ask ourselves, how do we approach this, was a completely new way of thinking. And my colleagues and I, it invigorated us. And it was amazing, the commonality and the uniqueness of the thoughts that we were having and going forward. Basically, machine learning responded to these things and the population structures and the markers and the numbers. By this time, we're really excited and we're now set on exploring. Now, ultimately, our goal is commercial products. So goal one, and the first thing that we do is we apply this information and get those top hybrids in. Our next project was a joint project between two breeding programs here. And this project generated 4.8 million hybrids. Now, that's an amazing amount of information to come back as a summarized result. But we had multiple growing regions, multiple types of agronomic practices and farming and such. But we also included in this second and third breeding cycles of the initial study. That is to say, here we are now pushing forward off of that initial base a number of years down and further up the genetic gain curve. And we did this through a pipeline integration. At this point in time, we've integrated this into our pipelines. And that was one of the goals from the beginning. How do we integrate this tool into a pipeline? And what we learned was astounding in this. One is, first and foremost, when you have multiple environments and multiple targets or targets that can vary, you need to develop variable commercial pathways to accommodate this. And if nothing else, it's to get your traits focused towards a target. Multiple opportunities is a really nice thing, but you need to understand them and prioritize them. It made us as breeders be more focused and disciplined with our clear prioritization of our commercial goals. But that's good. That is a very good thing. It makes us more efficient. The next generation of questions is always the key, though. That is that when we started to see our initial analysis, we hadn't asked the question, but what we saw was that certain genetics coming from either program tended to go one way or another. And some of them tended to go both directions. And now we needed to ask questions that help us understand this type of thing. Our relationship now, our team with the Computomics statisticians is a very close one. Getting them to understand our realm, what we're working, our goals, our measures, and what we're seeing and how can we address this now became paramount. It became fun in a sense. Those discussions were great. But now we also, besides the structure and the population, the inputs into the model building, we also have to communicate now the outputs so that they come in packages that we can unpack, deliver the top goal, the commercial potential hybrids to the field, and then start to understand that. So our evolution of thinking and the way we're approaching breeding and the integration of the pipeline is evolving quickly. We're on a learning curve by all means here. We're gaining confidence as well now. We've seen the accuracies of this, and now our expectations of what's going to the field are really very much up there. There's a point in which our thinking changes, and I have to go back to a movie classic, Dumb and Dumber. There's a scene in there where 1 in a million is used. In one sense, the party saying more like 1 in a million is meaning there's no chance in the world. On the other hand, the party receiving that is thinking, so you say there's a chance. And that's a really important pivot for a breeder and the breeding with the machine learning. 1 in a million goes from no chance to I've got 5 of these, I've got 500 of these. I can find them and I can get them to the field. And that level of thinking takes you into these dimensions. You're not only thinking about improvement within the pools on either side of it, but the interactions and the improvements in the hybrid combinations in all possible forms. 1 in a million is now a possibility.
Thank you, Keith. That's a great explanation of the technology. So obviously, it's completely changed your approach to breeding, and you now seem to routinely be able to define your breeding targets. Can you tell us a little bit more about that, please?
Yeah, sure. I mean, it's really interesting to be talking about this with you guys. We're not experts by any means, but what we are is we're becoming really good students in trying to understand the full potential of machine learning and its application in the commercial breeding program. So we're continuing to explore this. We're constantly coming up and discussing as a breeding group here the more challenging but very disciplined and structured population schemes. And we're doing this to start to try and answer these next generation of questions. It really has moved to a multi-dimensional chess game in the sense that we're looking at trying to identify, understand, and exploit. And that's kind of an odd word, but that's what we do as commercial. We exploit this knowledge and these genetics towards commercial products. But we're talking about genome interactions now. And not only within a pool on one side of the hybrid, a couple genomes here, but in multiple genomes in a hybrid combination. And so being able to start to think in those terms, now we're starting to try and define and understand the responses to environmental impacts. These challenges that every hybrid seeds in every location that are unique, and how those impact the interactions that we're trying to document is we're trying to basically find more efficient pathways of A to B commercial products. One of the projects we're currently working on is starting to address this. It's a multi-program breeding program project, again, so joint testing and joint breeding, joint genetics and such. But this is about the exploitation of the diversity and the depth of our models now. We have genetic umbrellas under which we've built models for performance prediction. And now we're trying to bring in environmental interactions in multiple breeding programs. That is to say, we're going to try to start to identify and define environments across a wide swath of the Corn Belt. This project generated now 581,890,000 virtual hybrids. That's big. Selection intensity. So we've learned from past, we've defined commercial tracks of these outputs of the data. We're looking at selection intensities within these commercial tracks of 0.001%. That is to say, 1/1000% for a selection intensity. That's an incredible thing to put in a genetic gain model. There are multiple commercial tracks, and the thing of it is what we've learned is we can predefine these so that we focus the results towards down these tracks, knowing that this is where they're going. And that's a very big change in breeding. You're not trying to find where this goes or what it does based on the data. It's you built it to go this direction, and then you can assess it and take the best ones and to meet that. The other thing that's changed is that the initial testing in the field that's not part of building a model, that has not grown, even though we've integrated more and more and we're generating more virtual hybrids. The new genotypes of these populations that we built are now the starting point for most of this, not the field. And the model is what puts this genotype into motion into a pipeline. And what you wake up and realize at one point is that as a breeder, I now have two pipelines. I have a virtual pipeline, which there are literally millions and millions. And I have the applied pipeline that's in the field that is destined for the farmer's field, the commercial product at the end of the day. It's a different world that we're living in. Most of our breeding centers around the development, advancement, and controlled expansion of our genetic umbrellas. We're working in controlled and structured. We're using our quantitative genetics training to basically be more efficient, get the right things in the right place. This is completely now building forward. Everything that we do and we go forward, what comes into our nurseries has a place, has a position, and is on a track going forward. We're more aware now than ever of our traits, the traits' heritability, the identity of the allelic identity in state versus identity by descent is crucial in keeping that accuracy of our model up. And basically our population coefficients of covariance. How can we keep our predictions high as we're broadening and exploiting this? And really now we're exploiting this information, this cumulative massive information. Machine learning really gives us two things. We get these performance predictions which we can apply directly. This is our main goal, commercial quality hybrids in the field. The accuracy and the efficiency of these hybrids to advance is incredible and has stayed that way. And that's has to be our main goal. The components, though, of machine learning builds these models through sheer power and the questions asked, but they'll list out these components of what's impacting the model most. They now become your quest of understanding and how to utilize and how to exploit those responses and such. Bringing the environment in now really complicates it, but the accuracy is really where we're going for.
Yeah, thank you. And I just sort of want to sort of touch on the actual incorporation of sort of environmental data. I think that was a multi-program project as well. So if you could just tell us a little bit more about how you incorporated the environmental data and how that impacted on your program, please.
Sure. I think I'll go from the top down, and one is that the first and foremost, the interaction between breeding programs and the exchange of ideas. I think that the utilization, they're getting into the machine learning and how to apply it has brought us closer together as a breeding team, and the exchange of ideas and how we can use leverage of our numbers together has really changed immediately. But the whole idea of the environment started basically as a discussion from one of the projects where we had environmental differences coming in, and the machine learning gave us indications that, wow, there was something there that would suggest that do we predict hybrids to specific hybrids to a specific location, or are we looking for hybrids that are stable across all? And it was a simple question, which you would think immediately you have your idea that, this is what we want. And everybody sort of had that until you realized that everybody had a different answer. And when we had the discussion about this, we found that there was no single answer that really was, that there were more questions that needed to be asked about this. So what is an environment? And what are the factors that define that environment become? In doing this, we came up with sort of an idea of the static and the dynamic factors. So static was something, well, if they side dress nitrogen here, or population density, or fungicide. And the dynamic was more of the weather related and soil types and this type of thing. And so what we found was that even those that went into the static were not really static, because timing of all these things, the agricultural practices would vary those. And then also those dynamic ones, well, a dynamic was a relative term, because what can you count on and what level? So we started to say, okay, well, what can we measure? Because we need to bring in some of these factors and such. And at this point in time, we decided, let's make a good decision. And we went to our Computomics statistician. And we said, here's what we're trying to do. Here's our goals. Here's the things that we're thinking of. What kind of data can you most effectively use to start to answer these questions and dig deep into these factors and these interactions and such? And so that was a great starting point. And again, what we decided was, let's start simple. Let's go with knowns, measures that we can count on, and measures that he can utilize in asking these questions. So in a sense, we use this type of discussion to define our testing scheme across the environments. We defined the measures that we could be used, and so that we were comfortable with them, and we could understand how they would be used in machine learning so that the results could be interpreted more cleanly. And then we had to fit this within our budget, because when you ask a statistician about how many we should put where and how many locations, he'll say, what I would really like is you put all genotypes at all locations across all environments. And then we have to laugh and say, okay, we don't have the budget for that. So reality versus accuracy becomes a balancing act here. So what we're doing, this project, we're looking for G by E, but each breeder wants to understand with their environments, what are the G by E components, the factors, and then at the same time, we want to broaden that to across our programs, what are the differences, and what are the genetics that can respond to these, that can go there. And the movement across environments of these genetics and those interactions, those responses, are now the key. We also needed to incorporate in this genetics that had the eliteness that could move across breeding programs and environments and had the capability, because again, our number one goal, commercial hybrids. So here we are now, the balancing act of accuracy versus reality and application. The short-term goals, hybrids that can be directed to specific environments, each environment. How do we capitalize on this and get commercial hybrids? And then hybrids that are stable across all environments. Our production people would love one hybrid to produce across environments as opposed to specific hybrids and type of thing. But understanding this and deploying them appropriately. The midterm goals of this are basically identify the key environmental factors, defining and identifying the genetic responses that make a positive. This enables our product to perform the way we do. What we realized then is that in essence, we're trying to build our pipeline to precision farming. And that's something that's really important. Here at Beck's, we have a tremendous group called PFR, Practical Farm Research. And for years and years, they've done major studies on new advancements in technologies and techniques and such to say, here's what a farmer can do in these circumstances to be better towards successful. We have agronomists out there that basically are trying to advise farmers on what's happening, what they need, what they can do to be more successful. And then I work very closely with our product placement guys whose job is to identify those elite hybrids and get them into those environments or those locations or regions where they'll do the best and be the most stable for the farmer. We have a platform called Farm Server, which takes all of this information and puts it at the farmer's fingertips so he can go to his farm and make his decisions on what he needs to do to be successful based on all this information. What we would like to do is start with the genetics right from the initial crosses and start to feed into this type of thing so that what we learn and what we're building is at the farmer's fingertips when he goes to make his choice for the hybrids he puts in his field.
Okay, thank you. That was an absolutely great overview of how you've utilized the technology within your program. So I think it just needs to summarize really. And so I just want to just really finish off by saying Exceed Score allows the breeder to define the traits with the goal of maximizing the heterability within the target environments. It provides all of the genetic information within the germplasm and identifies the top lines which are relative to target traits in all populations. This provides defined breeding pathways. The breeder can focus on population management and new line interregression, which maintains and increases genetic diversity whilst maintaining accuracy. The breeder can also identify elite combinations and new future donors, as well as understanding the interactions and effects of the genetics on the environment. And then just to finish off, I just want to show you how we at Computomics work with breeders. So we work with breeders throughout the whole of the breeding pipeline. We can offer advice on how to build robust data repository of both genotype and correlating phenotype data. We can help to optimize your phenotyping strategy by advising the breeder on which type of phenotypic measurements are optimal over specific timeframes. We can build a model that includes all of the target traits and then simulate all of the possible crosses so the breeder utilizes their field trials effectively with the best performers. The model is improved by genotyping the progeny and eliminating the unfavorables, and by advancing the most promising crosses, which provides genetic gain each cycle, as well as, of course, maintaining diversity. A location model can also be built so the breeder can select the best genotypes for a specific climate or location. There is also the ability for the breeder to breed for specific targets, as well as identifying causative genes for relevant traits. I've just mentioned a few of the applications here, but there are many, many more. So this concludes our webinar today, so I'll hand you back to Mark.
Thank you so much, Keith and Ruth, for that great presentation. Before we wrap up, we have some time to take questions. So please, if you have a question for our speakers, please type it into the chat box at any time. We have several questions that have come in from our audience, but I'm going to be a little bit selfish today, and I'm going to start off with a question that I'm curious about. Keith, what's the number one myth you encounter around machine learning?
Well, that's an interesting thing. Machine learning came in with such fanfare and all sorts of things about how you could it would tell you anything. And there's that sort of myth out there where you just generate a cloud of data and turn machine learning loose on it. And it can find things, no doubt about it. But what we're learning, we're still exploring, we've still not found the limits of what machine learning can do if it's given the right type of material and the right questions. And what we found is that machine learning is not an entity, it's a tool, a vehicle. And it's the questions and the setup to allow it to go and find these higher level interactions. It's being able to compile the complexity into answers that can be taken and applied. And I think that that's something that has an incredible wide area of operation, because it can be applied to almost anything where you're asking a question and wanting a complex simplified to a simple or applicable type of an answer. So I think the myth is that it's its own entity, when actually it's an incredible tool for the people who are asking the right questions and providing the right information.
Yeah, I guess just like any tool, it can do amazing things, but you have to know exactly how to use it and when, right? No, thanks, Keith. I have a bit of a technical question here. Are the cross predictions reported used independently, or are they groupable and clustered by commonalities, for example, by impacted genes, metabolic pathways, or phenotypic profiles?
As we got more complex in what we were doing and the opportunities to apply those, to exploit those, what we realized is that understanding and defining your traits and the trait packages of what do you want in that package. It's sort of when you talk about these type of hybrids and you go look at hybrids with the product placement group or the sales, does it have the look and performance? That is, what does the farmer want to see? If a farmer drives by his field early in the spring, what makes him feel comfortable and confident, even though he knows the season is just beginning? As he drives by that field later in the year, what does he want to see? Late in the season, as he's making his choices about how to approach harvest and such, how does he feel comfortable and anxious in a good way about it? But most importantly then, what's his yield meter telling him as it's coming out of the field? And so when you can start to address packages that incorporate the traits that meet those kind of criteria, and what you see with the different genetics, that diversity gives you different opportunities. And that was really understanding what we wanted and building it allowed us then to focus it and channel it into those types of things. There's no way in the world I'd want to receive a massive 581 million hybrid predictions without understanding what I was going to do with them and how. And so it's one of those learning curve things of using machine learning.
How would GE modified genes and traits affect your models and predictions?
It's a good question. I think that's what we're trying to learn more effectively. Initial indications would suggest that if you can find the genetic interactions of two genomes making a hybrid, that's one set of questions and what they can predict into a vanilla environment. And now what we're trying to chase down is those impacts, the key factors that the environment puts on the genetics, how that changes that set of interactions and where that fits in the expression. And that's a whole nother level of questions and interactions. And those are very high level, and those are where machine learning lives and thrives best.
Keith, in your opinion, what would be the main barriers that prevent breeders from understanding the advantages of this technology, and which would make it which would make it difficult to incorporate into a breeding program?
There's a period. I said early on, we started simple, and we wanted to provide a good genetic solid platform of genetics, not only and especially not specifically for the commercial aspect of things, but to understand the tool. And I think that that's the thing that I would say that you're going to have to learn to think differently, because you have more options and you have a different level of understanding. Some of the questions that we're asking, we never could ask before because we couldn't enter into this higher level, large number interaction set. And so there's a level of faith. The big expression that keeps coming up is you can't fully understand it until you experience it. When you do experience and you go, how can that be? The second time you experience it, you go, oh my, what have I done? And then the third time you experience it, what can't I do? And it is a real progression in understanding and believing. It changes the way you think.
This was addressed a little bit in our chat, and you touched on some of these things earlier, but what is the difference between machine learning and deep learning? Can you talk a little bit about that?
I'm not sure, to be honest with you. I'm not familiar with deep learning. And quite honestly, I'm learning about machine learning. So I'm afraid I don't really know in that sense.
There was a comment made that deep learning is one technique in the field of machine learning.
Yes.
So I guess it could be part and parcel. Ruth, do you have a little bit of insight there?
Yes, there are many sort of models or algorithms that you can deploy, and deep learning is one of those. But you know, as you all know, I'm not the technical person here. But it's something that we can ask one of our technical team and come back to you on. But yeah, it is, as far as I'm aware, another form of machine learning.
And I will highlight that Ruth's email is up on the screen. So if you do have any questions, please feel free to email Ruth directly at ruth.mays@computomics.com. I am just going to see what other questions we have come in from our audience here. What would be the minimum data set, population size, number of locations, etc., to train a machine learning model? Any insights there?
Yeah, so it very much depends on the species of the crop, and it is variable. You don't necessarily need an abundance of data, but you need robust data. So balanced data sets of good genotyping and correlating phenotype data is a good basis. And I guess you can start from around 500 lines upwards for specific species, but obviously more complex would need probably higher numbers.
Keith, obviously you touched a little on this a little bit earlier, but can you give us a quick snapshot? How can machine learning speed up a breeding program?
There's a number of ways, I think. Like I said, that initial list of hybrids coming off, the top ones going straight to the field. Those are nowadays, those are coming off of genotypes of lines that have really only been in the field as initial seedlings. And the next year they're in testing and type of things with expectations. And the accuracy, when the percentage of those advancing in a commercial fashion, that is against the commercial checks of interest, increases to a higher number, a significantly higher number, Numbers are good, especially in the closing phases, the end of testing and pre-commercial type of thing, because it gives you more for the environment to identify which ones can take it and such. I think the thing that's most unique, and I'm easily amused, but I still get a kick out of it, is I'll walk into my nursery with these new inbred lines that have never been to the field before, and to stop and to realize that I can actually walk to individual plants and say these two, and before they've even been replicated and increased and such, these two will do what I want them to do in the field, and getting forward. Starting new breeding cycles immediately. So generally speaking, you like to see the performance before you commit to the next generation and such. And now we're using the data to build immediately. So you can fast track things through various means. So is it speeding up? Yes. Our genetic gain is increased. So are we getting faster towards our ultimate goal of the genetic gain track? Yes. So machine learning has done it in those fashions.
What would your advice be to a computer science student who's just starting out in this field, what they should be learning? What would be your top piece of advice to them?
Well, that's an interesting one, and yeah, one really for the technical team and not for me. But we can certainly feed that question back.
Sure, and feel free to email Ruth, and she can direct you to the appropriate people. Are you mainly using supervised learning? What defines seed performance when real-world evidence is not available?
One of the things that we've done in integrating it into a pipeline, and I know my colleagues and I are big sticklers on this, is that don't get lost in the virtual world. We work very hard in breaking the data down and grounding it into the reality. And it becomes a part of the discipline of when we validate the study and the models, or when we make groups of hybrids to exploit and go forward. All of that then is brought back and grounded. Is it doing what we want it to do? Does it hit the criteria and such? So as I said before, what you understand very quickly is that you have two pipelines, the virtual one and the one that's in the field. The focus needs and the discipline needs to be kept that the one that matters is the one in the field. And it is the priority, and it needs to be in sync and make sense with the virtual hybrid. So, but we do a lot of things to ground all of this data back to the field. Does it really do what it says it should do?
Another very technical one I'm going to throw at you. Can machine learning correctly compensate for genotypic imputation, or can it suggest pockets of predictions where additional data could be beneficial?
All I can say about that one is that, yeah, it does impute genotypes. And again, it's quite a technical question, so I'm not the technical expert, but we can certainly provide answers for that. But we do impute genotypes for sure.
I guess I'm the applied guy, so I think that when we start to break the data down and we start to see indications of things happening, and it's not that doesn't answer the question exactly, but the fact that the ability to observe these things, to ask better questions and say, what if we did this? In the one project jump where we increased the number of progeny, or we tripled the number of markers, or we integressed genotypes to give it more possible combinations, what we saw there was that machine learning could utilize and capitalize on those additional things that we could put in. And so in a roundabout way, that the answer to that is yes. I mean, feeding machine learning is part of the process, giving it what it needs to flex its muscle.
How hard is it to switch and to commit resources to learning populations?
It's painful. And that's why we've really worked hard to build structures and trees to eliminate or minimize it. As I said before, introgressing and starting into new types of genetics or beginning new umbrellas is still about the same amount as we started with. And basically what we try and do is utilize the questions that we're asking and those different groups of hybrids that are basically on a commercial type trajectory as feedback based data for developing this. So you really, that had to be a breeder that asked that question. Those plots are painful plots because you really want everything on a trajectory for a commercial product.
Another technical one I'm going to throw at you here, which is better for machine learning, normally distributed variables or bi or trimodal distribution? Can you provide any insights there?
I can't give you any definitive answer, but what I've seen, because we are starting to incorporate different oligogenic versus quantitative type things, I think it really has the flexibility to be powerful in any of them. It's just the type of data and the structure that you provide that defines what those are and how it can use that information. I think there's an incredible amount of flexibility. It's just how you ask the questions and that what you provide is information for it to work with.
Before we sign off for the day, I'm going to ask you to gaze into your crystal ball for a moment. Where do you see this technology going in the future? Like, what's the one thing that right now might maybe sound like science fiction, but one day could potentially be possible?
That's a good question. That's a hard question in the fact that we've really tried to explore where the limits of machine learning are. And what we ended up with was more questions that we wanted to chase than we could chase. And I think that the ability of technologies like drone technology and the ability to measure wild things and organize clouds of data, one day I think that machine learning can be put onto tasks that seem insurmountable if they are asked to do it correctly. I think that machine learning is going to be part of the future because everything we do, from self-driving cars to breeding corn hybrids, we want to know where we're going and how to get there most efficiently. And if the intermediate, the helper is machine learning to understand those environments and how we need to navigate and to get us there safely and quickly, then I think that the application is endless.
Yeah, I suppose at one time even machine learning itself would have sounded like science fiction. And here we are today. I guess that's just how progress works. Well, that is all the time we have for today. If you have any other questions or would like more information, please just email ruthcomics.com. Her email is there in the bottom left-hand corner. Thank you, Ruth and Keith, for your time today.
Thank you.
Thank you very much.
And thank you to our audience for spending the last hour with us. We hope you found this information valuable. Again, this webinar will be available for viewing at seedworld.com following today's event. This is Mark Zinkowitz of SeedWorld signing off. Have a terrific day, everybody.